Dr. Chen is about to hit her eureka moment. After months and months of meticulously engineering a protein to calm overactive immune cells, it worked perfectly in all the lab tests. But it failed miserably when she injected this into bunnies. Their immune system spiked up instead of subsiding, and it's definitely safe to say that she's not getting that FDA approval anytime soon. The problem wasn't that her protein was designed badly or anything, it was because it was insidiously contaminated with a certain chemical. Dr. Chen isn't alone in this either. Many drugs for cancer, diabetes, and diseases like COVID-19 are proteins, making manufacturers pay tooth and nail just to simply detect and remove this protein. The industry is around $2.32 billion right now, and it's projected to reach $3 billion by 2030. But, perhaps, the most insidious thing about this toxin is how it's misled us. It casted doubt on entire research papers on COVID, HIV, dengue, and many other diseases, muddying our understanding and misleading follow-up research papers for years to come. So, what is this chemical? What makes it so insidious? And how do we even fix this problem? Chapter 1. Water Fever. In the early 1900s, injectable drugs became very popular. I mean, this is the century of both world wars, so injectables will be on very high demand. But doctors started noticing something very strange. Even when patients were given the right drug, they get spiked fevers, go into shock, and unfortunately die in some cases. People are getting high fevers all of a sudden, even when drug concoctions were made with distilled water. The medical community nearly believed that such a thing as water fever existed. The culprit? It wasn't the drug itself, but it came from bacterial contamination. Later, we found out that it is actually part of the cell walls of gram-negative bacteria, known as endotoxins. These still remain even when the bacteria dies, because they are essentially part of its corpse. At the time, we didn't know why endotoxins are toxic, but we knew that removing them certainly led to great results. Well, great, we just eliminate bacterial contamination from water, and we don't have any more endotoxins, and no more patients get sick because of our drugs. It looked like all is well and good, but more on that later. Rabbits were used to test for endotoxins constantly, until we later switched to a better method of using the blood of horseshoe crabs. In fact, every drug you've ever been injected with has to be tested for endotoxin using this method. These creatures are being sacrificed for our health and safety, so scientists are hard at work to find ethical ways of doing this. But that's a story for another video. Well, the problem of endotoxin is solved. For drugs. That are small molecules, that is. But many important drugs are big proteins, such as the cancer-hunting antibodies from a previous video. Endotoxins are very hard to remove from proteins. But why? What's so different about proteins, and what can we do about it? The answer lies in how endotoxins actually work. Chapter 2. Why do endotoxins work? In 1998, Professor Bruce A. Butler discovered new receptors on immune cells. These receptors are known as TLR4, and Professor Butler found out that endotoxins stick to and activate these receptors. His work is the final stroke in the painting that confirms that endotoxins are actually not toxic in and of themselves, but they are toxic because of the way our immune system reacts to it. This isn't a bug in our biology, it's a feature, an ancient overprotective one. Even trace amounts of endotoxin activate a strong immune response. When your body detects even a single smidge of bacteria, it's taken out with overwhelming force. But this can overreact when bacteria floods your system, like in sepsis, leading to fever, shock, and even death in unfortunate cases. But all in all, we now know why endotoxins work, and this has big implications. First of all, it changed the way we thought about our immune system. At the time, the first line of defense was thought to be a non-specific, no-brainer attack anything it sees type of immune system. Now we know that they can specifically target bacteria and other harmful things using these TLR receptors and send out specific signals to activate other parts of our immune system. This was why Butler was co-awarded the Nobel Prize in 2011. This discovery was great for the medical community. But it unintentionally led to a slew of misinformation in the scientific community, which brings me to the next implication. Everyone was trying to test what activated TLR4 because they want to fully understand how diseases activate our immune response. But the deeper we studied TLR4, the stranger things got. It started picking up signals from proteins that should not have triggered any immune response at all. Things like proteins that help other proteins fold. The problem still existed today. How and whether the COVID spike protein, dengue viral proteins, and HIV activates TLR4 or not is still contested in this 2023 paper. They're quite literally known as dirty little secrets by the industry. So, were these proteins dangerous? Or were they just dirty? And first of all, why would endotoxins even be associated with proteins at all? As you've just seen, even if the protein doesn't come from bacteria, they still have this problem. The answer has to do with how scientists prepare proteins in a lab. The cell of every living organism makes proteins to do almost every task. The cheapest and easiest cell to use for this task is E. coli, a gram-negative bacteria. You know, the source of endotoxins. But can't we just purify the endotoxins after making the protein? How hard could this be? And this is kind of where the insidiousness of endotoxins come in. They stick very well to proteins, especially if they're hydrophobic membrane proteins. They've also been shown to stick very well to protein purification tags. Worst of all, they stick to purification devices. More on why that's worse than it sounds later. Since they stick to proteins, they can disrupt protein folding, cause protein aggregation, and reduce protein bioactivity. Not only do they poison you, they make your drugs and assays worse. In labs, scientists actually reuse a lot of equipment, including purification columns, the very columns that were tainted with the endotoxin from the previous person. This basically causes a lab-wide contamination, muddying our results and stammering our progress. Huge problem, right? So how do we stop that? Chapter 3. Clearing contamination. We exploit the innate properties of endotoxin. It's made of sugars on one end with two negative charges and a lipid on the other. That makes it very similar to soap. It's okay with both water and oil. So, one way we can go about this is we find a protein that really loves that particular tail more than any other protein. This protein is called polymyxin B. There are other methods too. Some exploit the negative charges, some use their dual polar nonpolar nature, others use filter with tiny holes. But there's a problem with some of these approaches. Every time you try to purify or filter out things, you inevitably lose some products along the way. This is okay for research, but it's terrible for manufacturers. So, we do everything in our power to remove endotoxins before they even enter our labs. It comes down to choices as subtle as what plastic container we use to handle chemicals. Some materials like polystyrene and polycarbonate aren't as sticky to endotoxins as polypropylene. In general, hydrophobic materials will make it harder to wash off endotoxins, making them stick with your experiment for longer. While hydrophilic ones align the endotoxin such that it's super easy to wash off. Not only that, we can just avoid using E. coli entirely. We use dedicated mammalian cell lines. This not only eliminates the primary source of endotoxins, but the components of the mammalian cells also allow human proteins to fold in a more human-like way, making it more accurate for us humans. This and other design choices, significant and subtle alike, that we made at our dedicated production at Sinobiological allowed us to minimize endotoxin contamination to 100 times below the current industry standard. Be sure to learn more about that on our website. Endotoxins have been tripping science up for a century, hiding in our work, clouding our discoveries, and even muddying entire fields of research. But every time we looked deeper, we learned more. First, the bacteria left something behind. Then our immune system was the reason it was toxic. And finally, even our own experiments could be misled by it. Each level of understanding didn't just give us better ways to fix the problem, it also revealed how insidious the problem was. That's the lesson of endotoxins. The deeper you go, the more you see. And only then can you really start to fix what's broken. Thank you immensely for watching.